Hi, my name is Clement Savage and I'm doing biology at AS level, and today I'm going to be looking at DNA replication and protein synthesis. So it starts off with self-replication. DNA can copy itself, and it copies itself before cell division so that each cell has the same full amount of DNA. It starts off with the hydrogen bonds between the two polynucleotide strands. They break, so therefore the helix unzips to form two single strands, and the hydrogen bonds break between like guanine, a cytosine, an adenine, a thymine, so that's where they break. And then secondly, each original single strand acts as a template for a new strand, and these free floating DNA nucleotides join onto the exposed bases on each original template strand by complementary base pairing, which is basically just adenine with thymine, cytosine with guanine. They go all together. The bases just match up using this complementary pairing. The nucleotides on the new strand are joined together by the enzyme DNA polymerase, which is spelled Polymerase, polymerase. And hydrogen bonds form between these bases on the original and new strand. Lastly, each new DNA molecule contains one strand from the original DNA molecule and one new strand, but they come together to form a DNA molecule and are all twisty. And this type of copying is called semi-conservative replication, because half of the new strands of DNA are from the original piece of DNA. So that is self-replication, and now we're going to look at the fact that DNA contains genes which are instructions for proteins. A gene is a sequence of DNA nucleotides that codes for a protein, a polypeptide. And proteins are made from amino acids. Different proteins have a different number and order of amino acids, and it's the order of the nucleotide bases in a gene that determines the order of amino acids in a particular protein. And each amino acid is coded for by a sequence of 3 bases in a gene, and the different sequence of base codes for a different amino acids. So I have an example in my notes in my book here, but it's kind of hard to explain. Like, for example, in the DNA, if the order of the bases on the DNA was GTC, 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 the amino acid would be valine, or if it was TCA, which stands for thymine, cytosine, adenine, TCA, 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 it would be serine. So yeah, I'll do one more, which is protein synthesis. And DNA is copied into RNA for protein synthesis, and this is because all the reactions and processes in living things need proteins, and DNA carries the instructions to make proteins as genes, and that's found in the nucleus. The organelles that make proteins, like ribosomes, are found in the cytoplasm, but the DNA molecules are much too large to move out of the ribosomes. Out of the nucleus, sorry. I'll say that again. The organelles made of proteins, the ribosomes, are found in the cytoplasm, but the DNA molecules are much too large to move out of the nucleus. So, to combat this problem, sections of DNA are copied into RNA, and it's the RNA that leaves the nucleus, therefore, and joins with the ribosome in the cytoplasm, and therefore it can be synthesize a protein. So DNA and RNA are vital for living organisms to produce proteins in order to grow and develop. So that was DNA replication and protein synthesis. Good luck with your exam in a few weeks.